Hello everyone, myself Vaishnavi Mane from SYBSC roll number 6229. My topic is Translation Step 2 Elongation. In this video, we will see how elongation of the polypeptide chain in translation takes place. Elongation is the stage where the amino acids are brought together by forming peptide bonds and make proteins. So now let's see how elongation takes place. The factors that aid in the elongation are called as elongation factors. There are three elongation factors. They are: first is EFTU, that is elongation factor Tu, which helps in the addition of amino acids. Second is the EFTS, that is elongation factor Ts, which helps in the recycling of the EFTU. Third is the EFG, that is elongation factor G, that helps to translocate the tRNA. This is the 70S initiation complex formed in the first step of translation, that is initiation. Here, the 30S subunit and the 50S subunit of the ribosome are bound together with the initiator tRNA charged with amino acid formylmethionine at the P site of the 30S subunit where the start codon is present. This is the EFTU-TS exchange cycle where the EFTU is recycled by the EFTS. First, a second elongation factor, EFTS, binds to the EFTU and displaces GDP. Next, the GTP binds to the EFTU-EFTS complex to produce an EFTU-GTP complex simultaneously with the release of EFTS. An aminoacyl-tRNA binds to the EFTU-GTP and that complex can now bind to the A site in the ribosome where the appropriate codon is exposed. In the next step, the charged tRNA binds to the A site with the help of EFTU. The EFTU is then released from the A site when the binding of the charged tRNA is complete. The ribosome maintains the two aminoacyl-tRNAs in the P and A sites in the correct positions so that a peptide bond can form between the two amino acids. Two steps are involved in the formation of this peptide bond. First, the bond between the amino acid and the tRNA in the P site is cleaved. In this case, the breakage is between the formylmethionine and its tRNA. Second, the peptide bond is formed between the formylmethionine and the serine attached to the tRNA in the A site in a reaction catalyzed by peptidyl transferase enzyme. Translocation. In this step of elongation cycle, the ribosome moves one codon along with the mRNA towards the 3' end. In prokaryotes, the translocation requires the activity of another protein elongation factor, that is EFG. An EFG-GTP complex binds to the ribosome. GTP is hydrolyzed and translocation of the ribosome occurs along with the displacement of the uncharged tRNA away from the P site to the E site. It is possible that GTP hydrolysis changes the structure of EFG, which facilitates the translocation event. Then the uncharged tRNA at the E site of the 30S subunit is released. This requires GTP hydrolysis by the EFG GTP complex. Now we have an empty E site and an empty A site. The next aminoacyl-tRNA binds to the A site and thus elongation continues. The elongation cycle repeats until the stop codon is encountered. Once the stop codon is encountered, the elongation stops and the termination begins. This was all about translation step 2 elongation. Thank you for watching.